Hello dear students, today our topic of deliberation is microbial spoilage of foods. The main objectives of today's deliberation are growth of microorganisms in relation to food spoilage, identification of different types of microorganisms responsible for food spoilage, and various types of food spoilage. Food spoilage can be considered as any change which renders a product unacceptable for human consumption. Foods are not only nutritious to consumers but are also excellent source of nutrients for microbial growth. Microorganisms are the most important cause but not the only cause of spoilage of foods. Microorganisms can be used to transform raw foods into fermented delights including yogurt, cheese, sausages, pickles, and other alcoholic products. On the other hand, microorganisms can degrade food quality and lead to spoilage. Importantly, foods also can act as vehicles for disease transmission and thus detection and control of pathogens and spoilage organisms are important areas of food microbiology. During the entire sequence of food handling from the producer to the final consumer, microorganisms can affect food quality and human health. Growth of microorganisms in foods. Foods, because they are nutrient rich, are excellent environments for the growth of microorganisms. Microbial growth is controlled by factors related to the food itself, called intrinsic factors, and also to the environment where the food is stored, described as extrinsic factors. The intrinsic or food related factors include pH, moisture content, water availability, oxidation reduction potential, physical structure of the food, available nutrients, and the possible presence of natural antimicrobial agents. Extrinsic factors include temperature, relative humidity, gases like carbon dioxide, oxygen present, and the types and numbers of microorganisms present in the food. Since foods are of plant and animal origin, so it's worthwhile to consider those characteristics of plant and animal tissues that affect microbial growth. The plants and animals that serve as food sources have all evolved mechanisms of defense against the invasion and proliferation of microorganisms, and some of these remain in effect in fresh foods. Knowledge of sources of microorganisms and factors influencing microbial growth is necessary for food microbiologists because desirable growth conditions are needed for applicability of microorganisms for fermentation and single-cell protein production, and understanding of undesirable conditions are used for food preservation. Microorganisms and food spoilage. Food spoilage can be defined as any change in the visual, smell, and texture of food that makes it unacceptable for consumption, or the process in which food deteriorates to the point that it is not edible to humans or its quality of edibility becomes reduced. Food spoilage can be the result of insect damage, physical injury, enzymatic degradation, and microbial activity. Classification of foods by ease of spoilage. Foods are classified into three groups based on ease of their spoilage: relative stable or non-perishable foods, foods that do not spoil unless handled carelessly, for example, grains, flour, sugar, pulses, etc. Protectable or semi-perishable foods, foods those remain unspoiled for a fairly long period if properly handled and stored, for example, potatoes, apples, and onions. Perishable foods, foods that spoil readily unless special preservative methods are used, for example, milk, eggs, meat, fish, poultry, most fruits and vegetables. Types of food spoilage. Foods are classified into three groups based on the type of their spoilage. Physical spoilage, spoilage due to the loss or gain of moisture. Chemical spoilage, foods that are spoiled due to some chemical reactions of foods, for example, oxidation of fats, browning of fruits, and vegetables, etc. Microbial spoilage. Spoilage due to the growth and activity of microorganisms. The microorganisms responsible for food spoilage are molds, yeast, and bacteria. Now, molds. Molds are in the form of threads developed on perishable foods and are easily visible to the eye. They contain spores which can spread through the air and start new mold plant. When these molds find a favorable environment, they germinate and produce a fluffy growth, often white or grey, but sometimes bluish, green, red, orange, or some other color, depending upon the variety of the mold. Most molds are not harmful. A relatively small proportion of the molds found on foods are capable of producing toxic materials known as mycotoxins, of which aflatoxin is an example. Now, dear students, let's talk about yeasts. Yeasts are tiny organisms which are not visible to the naked eye, but which can be seen through the microscope. They multiply very fast and cause fermentation by acting on certain components of perishable foods like fruit juice, syrups, etc. During yeast fermentation, the sugars present in the food are broken up to form alcohol and carbon dioxide. Foods liable to be spoiled by yeasts are fruit juice, syrups, molasses, honey, jams, and jellies. Bacteria. Bacteria are unicellular organisms and are much smaller in size than either yeasts or molds. They occur in different sizes and shapes and are classified as coccus, bacilli, or spirally on the basis of their shape as seen under the microscope. They also vary in their requirement for food, moisture, acidity, temperature, and oxygen. Bacteria can grow and develop rapidly between 20 degrees Celsius and 53 degrees Celsius. Bacteria are classified according to the temperature range that they need for growth. A higher temperature than 45 degrees Celsius are known as thermopile, example in canning industry and milk processing plants. Temperatures between 20 to 25 degrees Celsius are called mesophiles, while as temperatures less than 20 degrees Celsius are called psychrophiles, for example in the refrigerator and cold storages. Dear students, let's talk about spoilage of different types of foods. Meat spoilage, sources of contamination, cutting board contamination, conveyor belts, temperature, delay between storage and distribution, fecal contamination from intestines. Storage temperature is the single most important control factor for meat spoilage. Several genera of molds grow on the surface of meat and can cause spoilage like Penicillium, Mucor, Cladosporium alternaria, but cannot grow on meat stored below 5 degrees Celsius. Meat spoilage is characterized by the appearance of odors and slime, which manifest when surface loads exceed 107 CFU per centimeter square. The slime is due to the accumulation of bacterial cells. Interestingly, meat spoilage, including poultry and fish, occurs without any significant breakdown of the primary protein structure. Instead, spoilage bacteria utilize glucose, free amino acids, or other simple nitrogenous compounds to attain population of about 108 CFU per centimeter square, at which point the organoleptic quality of the meat will clearly reveal it is spoiled. Now coming to the whole meats. The microflora of fresh whole meat is primarily composed of gram-negative aerobic rods such as Pseudomonas species, P. fragile, P. fluorescens, Acinetobacter, and Psychrobacter immobilis. Bacillus and Clostridia, for example Clostridium perfringens, are also common on all types of meat. Although subsurface portions of meat are generally sterile, some parts such as lymph nodes may be heavily contaminated. Mechanical disruption of the tissue during processing can distribute microorganisms from the meat surface throughout the product. Ground meats. Ground meats contains same microorganisms as whole meats but always have higher microbial loads because it has greater surface area which gives microbes better access to the food and also traps air to favor the growth of gram-negative bacteria, aerobic bacteria like Pseudomonas species. Every handling or processing such as storage utensils, cutting knives, grinders, setup can contribute additional contamination to the final product. One heavily contaminated piece, example a lymph node, can contaminate an entire lot when they are ground together. Vacuum packaged meats. During vacuum packaging, it has been found that not all the oxygen is removed, which is eventually consumed by aerobic microorganisms and the tissue itself. The consumed oxygen results in increased carbon dioxide levels and thus vacuum packaged meat gets a longer shelf life. The microflora shifts from predominantly gram-negative aerobes to gram-positive anaerobes and microaerophilic lactic acid bacteria like Lactobacillus, Carnobacterium and Leuconostoc. If nitrites have been added to the vacuum packaged meat, for example, to inhibit Clostridium botulinum in hams and bacon, lab domination is even more pronounced. In general, vacuum packaged meats are considered very safe foods and free from most pathogenic species of bacteria with the possible exception of Staphylococcus aureus and Yersinia enterocolitica. Spoilage in vacuum packaged meats is manifested by slime development due to overgrowth of microbes, greening caused by microbial production of hydrogen peroxide and hydrogen sulfide. Hydrogen peroxide production in meat has been associated with several types of lactic acid bacteria, primarily Lactobacillus. The oxidant, that is the hydrogen peroxide, reacts with nitroso-hemochrome cured meat colored compound to form green porphyrin compound. H2S reacts with myoglobin to form sulf-myoglobin in meats with a pH above 6. H2S is mainly produced by Schwannella petrifications and Pseudomonas species. Off-odor results from the release of short-chain fatty acids, the production of volatile compounds like acetone, diacetyl and H2S. The type of spoilage bacteria that will dominate is influenced by several factors that include: is the meat product raw or cooked? Cooked products have a higher pH which may allow growth of gram-negative facultative anaerobic pathogens like Yersinia enterocolitica. Raw products have a pH of about 5.6 which favors lactic acid bacteria, especially Lactobacillus, Carnobacterium and Leuconostoc. Nitrite concentration in meat. High nitrite concentration favors lactic acid bacteria. Low nitrite levels may allow growth of Brochothrix, Thermofacta, gram-positive rod, facultative anaerobic growth at 0 to 30 degrees Celsius with pH from 5 to 9. B. Thermofacta, an important spoilage bacterium in anaerobically stored meats kept at low temperature, but the bacterium is inhibited by nitrite. Now coming to processed meats. These products are composed of a variety of blended ingredients, any of which can contribute microorganisms to food. Yeasts and bacteria are the most common causes of spoilage, which is usually apparent in three ways. Slimy spoilage. Like other meat products, this occurs on the surface and is caused by the buildup of cells by yeasts, lactobacilli, enterococci or Brochothrix thermofacta. Washing the slime off with hot water can restore the product quality. Sour spoilage results from the growth of lactic acid bacteria which originate from contaminated ingredients like milk solids under the casing. These organisms ferment lactose and other CHOs in the product and produce organic acid. Taste is adversely affected, but the product is not harmful if eaten. Greening due to hydrogen peroxide or hydrogen sulfide production. Because greening indicates more extensive product breakdown, it's not recommended eating green wieners. Now let's discuss poultry and egg spoilage. General trends are the same as other fresh meats. Whole birds have lower counts than cut up parts. Additional processing steps add to the microbial load. When poultry is in the advanced stage of spoilage, the skin will often fluoresce under UV because so many fluorescent pseudomonas are present. Off odors generally appear before sliminess develops. The same bacteria can produce visceral taint, a condition manifested by off odor in the abdominal cavity or poultry. Eggs. Eggs have several intrinsic parameters which help to protect the nutrient-rich yolk from microbial attack. These include the shell and associated membranes as well as lysozyme and a high pH in the white. Freshly laid eggs are generally sterile but soon become contaminated with numerous genera of bacteria. Eventually, these microorganisms will penetrate the eggshell and spoilage will occur. Pseudomonas are common spoilage agents, but molds like Penicillium and Cladosporium sometimes grow in the ear sac and spoil the egg. Now let's discuss fish spoilage. Fish of both freshwater and saltwater contain high levels of proteins and nitrogenous constituents with low fat content and practically no carbohydrates. The microbial quality of fish is heavily influenced by the quality of water from which they were harvested. Fresh fish and iced fish are spoiled by bacteria, while salted and dried fish are spoiled by fungi. Bacteria on fresh fish are concentrated on the outer slime, gills and intestine. Most susceptible part of the fish to spoilage is the gill region, and the best way to detect spoilage in fresh fish is to sniff this area for off odors produced by Pseudomonas and Acinetobacter moraxella bacteria. The odors include ammonia, triethylamine, H2S and other compounds. Spoilage of crustaceans like shrimps, lobsters, crabs and crayfish is similar, but these products have some CHO and more free amino acids, so spoilage can occur more rapidly. Mollusks like oysters, clams, mussels, squid and scallops have more CHO and less nitrogen than either fish or shellfish. Microflora of mollusks can vary a great deal depending on the quality of water from which they were harvested. Spoilage of milk and dairy products. Milk is a very rich medium for microorganisms. Raw milk flora may include all important microorganisms found on the cow hide, which include soil and fecal bacteria, udder and milking utensils. It can include gram-negative, gram-positive, yeasts and molds. Psychotropic pseudomonas are common in bulk stored raw milk, which produce heat-stable enzymes that can reduce milk quality and shelf life. Pasteurization kills most gram-negative bacteria, yeasts and molds, but some gram-negative enzymes, thermotolerant gram-positive bacteria and spores survive. Pasteurized fluid milk is spoiled by a variety of bacteria, yeasts and molds. Pasteurized milk is more frequently spoiled by aerobic spore formers such as Bacillus, whose proteolytic enzymes cause curdling. Molds may grow on the surface of spoiled milk, but the product is usually discarded before this occurs. Butter has a high lipid content, thus making it more susceptible to surface mold growth than to bacterial spoilage. Some pseudomonas can be a problem. Surface taint, putrid smell caused by the production of organic acids, especially from pea putrefacients. Rancidity due to butter fat, lipolysis caused by P. fragi are common. Cottage cheese. Cottage cheese can be spoiled by yeasts, molds and bacteria. The most common bacterial spoilage is slimy curd caused by alkaligens species. Gram-negative aerobic rod bound in soil, water and intestinal tract of vertebrates. Penicillium, mucor and other fungi also grow well on the cottage cheese and impart stale or yeasty flavors. Ripened cheese. Ripened cheese with their low pH and high salt inhibit most spoilage microorganisms except surface mold growth. Spores of C. butyricum, C. sporogenes and others can germinate in cheeses, for example Swiss cheese, with intrinsic properties that are less inhibitory, for example low salt and higher pH. Spoilage of vegetables and fruits. Vegetables are a good substrate for yeasts, molds or bacteria. Typical composition of vegetables: 88% water, 8.6% carbohydrates, which includes readily available mono and disaccharides like glucose and maltose, as well as more complex oligosaccharides which are available to fewer types of microorganisms. 1.9% protein, 0.3% fat, 0.84% minerals, also contain fat and water-soluble vitamins and nucleic acids less than 1%. pH of most vegetables is around 6, within the growth range of many bacteria. Microflora of vegetables is primarily composed of gram-positive bacteria like lactic acid bacteria, for example Leuconostoc, Lactobacilli, Streptococci, Corniforms and Staphylococci, the latter coming from the hands of employees during processing. Staphylococci are usually unable to proliferate, but cross-contamination can introduce them into other foods where growth conditions are more favorable. Soft rot. One of the most common types of bacterial spoilage is soft rot, which is caused by Erwinia carotivora and sometimes by Pseudomonas species which grow at 4 degrees Celsius. Softening can also be caused by endogenous enzymes. Flavor sour story. Polygalacturonase hydrolyzes 1,4-glycosidic bond in pectin which leads to softening. Calgene made antisense RNA to tomato PG, thus delays softening in tomato, which can now be harvested after they are ripe. It was first commercially available genetically engineered vegetable. Now let's discuss mold spoilage. In vegetables where bacteria growth is not favored, for example low pH, molds are the principal spoilage agents. Most molds must invade plant tissues through a surface wound such as a bruise or a crack. Spores are frequently deposited at these sites by insects like Drosophila melanogaster, the common fruit fly. Other molds like Botrytis cinerea, which causes grey mold rot on a variety of vegetables, are able to penetrate fruit or vegetable skin on their own. Fruits. Like vegetables, fruits are nutrients-rich substrates, but the pH of fruits does not favor bacterial growth. As a result, yeasts and molds are more important than bacteria in the spoilage of fruits. Several genera of yeasts can be found on a fruit. Because these organisms grow faster than molds, yeasts often initiate fruit spoilage. Molds then finish the job by degrading complex polysaccharides in cell walls and grands. Average composition of the fruits. It includes 85% water, 13% carbohydrates, 0.9% protein, 0.5% fat, 0.5% ash, trace amounts of vitamins, nucleotides, etc., less water and more carbohydrates than vegetables, low pH of about 1.8 to 5.6. Specific spoilage organisms of fruits: blue rot, penicillium and fruits, downy mildews like Phytophthora, large masses of mycelium in grapes, black rot like aspergillus in onions, sour rot like Geotrichum candidum in grapes, cereal and bakery goods. These products are characterized by a low water activity which, when stored properly under low humidity, restricts all microorganisms except molds. Rhizopus stolonifer is the common bread mold, and other species from this genus spoil cereals and other baked goods. Refrigerated frozen duff products have more water and can be spoiled by lactic acid bacteria. Fermented foods and beverages. The low pH or ethanol content of these products does not allow growth of pathogens, but spoilage can occur. Beer and wine can be spoiled by yeasts and bacteria. Bacteria involved are primarily lactic acid bacteria like Lactobacilli and Pediococcus species, and under aerobic conditions like acetic acid bacteria, Acetobacter and Gluconobacter species. Acetic acid bacteria convert ethanol to acetic acid in the presence of oxygen. The anaerobic bacterium Megacephera cerevisiae can also spoil beer by producing isovaleric acid and hydrogen sulfide. Beer. Spoilage in packaged beer is often due to growth of the yeast Saccharomyces diastaticus, which grows on dextrins that brewers' yeast cannot utilize. In either case, spoilage by yeasts results in the development of turbidity of flavor and odors. Wines. Candida valdea is the most important spoilage yeast in wine. It can also be spoiled by lactic acid bacteria, which are able to convert malic acid to lactic acid. This reduces the acidity of the wine and adversely affects wine flavor. In some areas, for example Northwest, wine grapes have too much malic acid, so this fermentation is deliberately used to reduce the acidity of grape juice that will be used for wine. Fermented vegetables. Yeasts, molds and lactic acid bacteria can also spoil fermented vegetables such as sauerkraut and pickles, as well as other acid foods like salad dressings. Spoilage in fermented vegetables is often manifested by off odors or change in the color, chromogenic colony growth or texture softening of the product. In salad dressings, the first signs of spoilage are usually off odors and emulsion separation. Dear learners, this was all about today's program. Hope you have learned some new exciting things. Thank you for your attention. Have a nice day. Goodbye.